Okay, I think this much is clear to everyone. Now, next thing which I want to tell you is about the central dogma. So the process, central dogma is the basic process which takes place in our cell, and it is the sequential thing in which DNA forms mRNA and mRNA forms protein. As you can see in this diagram, this process is replication. Semi-conservative mode of replication is the way how the DNA replicates. Okay. So here what happens? The parent DNA unwinds and a new DNA is formed. So this is replication. By the process of replication, DNA forms a copy of itself. Now the DNA which is formed is unfolding again to form the RNA. Which RNA? The messenger RNA, by the process of transcription. Clear? Now this messenger RNA leaves the nucleus and goes into the cytoplasm. Here you can see the two subunits of ribosome. These two subunits of ribosome come together at the time of protein synthesis, and you can see the green colored here. This is the green colored messenger RNA, and this is the purple colored protein which is being formed by reading out the messages of the messenger RNA and then translating it into the language of protein by addition of amino acids, and this whole thing is being done in the ribosome subunits. So the central dogma is the process of formation of it's a unidirectional flow of information from DNA to RNA and from RNA to protein. Okay, so this is a unidirectional flow of information from DNA, where DNA forms messenger RNA and messenger RNA forms protein. Clear to all of you? Okay. Now, one more important thing I would like to tell you here is the mRNA which is produced here after the transcription, after the here the freshly prepared messenger RNA is very juvenile and very unstable, because there are so many enzymes present in the cell. So certain enzymes try to try to break down the messenger RNA. So to get it protected, what we do, we protect this long chain from both the sides. We protect the messenger RNA from both the ends. If it is a long chain, it has two open ends. See here, one end this side, one end this side. So one we call head and another we call tail. So here, to protect it from both the sides, we are adding a cap and a tail. So what we are actually doing, we are making certain changes in the messenger RNA, not inside the sequence, but before and after that particular sequence, to make it more stable and to protect it from the action of the exonuclease. Exonuclease, you understand, which exonuclease are the digestive enzymes. Endonuclease produced next here in the center, but exonuclease can digest it from the edges. So from the exonuclease activity, we need to protect the messenger RNA. And what has to be done? The freshly prepared, freshly synthesized messenger RNA has to be processed. And this processing is known as maturation of messenger RNA. Okay, so this maturation of messenger RNA is very much important so that it is safe when it moves around in the cytoplasm and is not being attacked by exonuclease enzymes or other things which can digest the mRNA. So that shall also be told in this particular chapter, but first we will be going with the transcription and then stabilizing the RNA and then the translation. Okay, so I think this much concept is clear to everyone. The central dogma, that means formation of protein from RNA, and RNA is synthesized from DNA. So unidirectional flow of information from DNA to RNA and RNA to protein is called as central dogma of life. Clear? I hope you are reading the notes which I have provided. Just read the notes properly, and from the next class onwards we shall be discussing about transcription in detail, stepwise and detailed description of transcription we will start from the next class onwards. Okay, thank you so much. All the best.